There's a lot of interest these days in linking genotype to phenotype, and most of the examples of studies like this come from organisms that we can breed and create F1s or F2s and map traits through genetic mapping. What I'd like to try to convince you of today is the possibility of mapping genes via comparative genomics, and we've seen some interesting examples of this from the mammalian literature. And so I'll first talk about a approach to looking at the origin of novel traits, in this case the origin of feathers, by looking at genomic elements that arise coincidentally with feathers on a phylogeny of vertebrates. And then second, we'll talk about the use of convergent traits to look at genomic elements that are arising convergently on those lineages exhibiting a particular trait. We're not as far along on this particular part of the project, but I hope to map out for you our general approach. So I'm going to also be focusing a lot on the sort of non-coding part of the genome. As most of you know, the genome of vertebrates is, the vast majority of it is non-coding. And so we hope to develop approaches that can monitor the important role of non-coding DNA as a regulatory network, as well as the coding regions themselves. In particular, we're going to talk about a class of elements called conserved non-exonic elements. These are elements that don't occur within genes, but are highly conserved nonetheless. You can see here this alignment going from mammals all the way down to zebrafish. This element here is about 2 kb, highly conserved. These are very likely to be regulatory elements that are influencing the expression of nearby genes. Now, feathers have been very well studied in the last 20 years. Their evolutionary history is exquisitely known. Paleontologists can even tell us the color of feathers of long extinct lineages. And they've also been studied from a developmental standpoint. And so we know a lot of the candidate genes that developmental biologists have used to study feather development. And so what we did was to survey the literature and track down almost 200 genes that were implicated in feather development. And we used a variety of comparative genomics tools to align the genomes of 19 vertebrates spanning birds to fish. And we then used a hidden Markov model to call these conserved non-exonic elements. These are similar to ultra-conserved elements, except they're not ultra-conserved. They simply evolve more slowly than a putatively neutral rate, such as the rate of fourfold degenerate sites in proteins. We discovered over 600,000 of these conserved non-exonic elements, 13,000 of which were close to and putatively associated with genes involved with feather development. So what we're going to do is essentially monitor the origin of the genes and the conserved regulatory elements themselves on the vertebrate tree. This is an example of a landscape where you have a gene involved with feather development, S-HOX. You can see how its exons are conserved throughout the vertebrate tree, and a variety of non-coding conserved non-exonic elements that are here color-coded according to their node of origin. So you can see the avian elements are in red, reptiles in green, amniotes in brown, and so on. And so we can sort of track the rise and fall of these elements using a variety of statistical tools. Now, our sort of proof of concept was to look at the beta-carotene genes themselves. These are well known to have undergone a burst of evolution on the branch leading to modern birds, and we detect that using our methods. By contrast, the regulatory elements don't seem to be doing very much in this, for this class of genes. We can find the 2 different peaks that correspond to 2 different clusters on different chicken chromosomes. Now, the really interesting part was the genes that were non-keratin, but nonetheless associated with feathers. What you can see here is that most of the genes that are associated with feather development, in fact, are very old. We can find these genes in humans, we can find them in fish, and they generally, we see very little innovation at the genic level beyond the Archosaurian node, okay? Now, by contrast, the regulatory elements here in blue, you can see they are arising at a disproportionate rate, peaking in the lineage leading to amniotes, and then still a highly extended region of innovation going all the way through to birds. And so we actually are proposing that the origin of feathers, aside from the beta carotenes here, the origin of feathers is much more an issue of regulatory innovation rather than the origin of new novel protein coding regions. This is just another way of showing the same data. You can see these three tracks going along the backbone of this tree, color-coded with red being rates of high innovation and blue being slow rates. We estimated that the non-avian dinosaurs, many of which sported protofeathers, had the full complement of protein coding genes required to make a feather, and approximately 86% of the non-coding regulatory elements. Finally, we use these approaches to try to figure out what kinds of things make birds unique. So we were looking specifically on the branch leading to modern birds and asking, where do we see a lot of regulatory innovation across the genome? And what we found was a dramatic peak near these two genes, insulin growth factor binding protein 2 and 5, which are involved with various aspects of body size change as well as limb development. This peak here shows that regulatory elements in the region of these genes are rising at a rate over 500 times the background rate in the genome. So this is a nice way of generating a hypothesis as to looking at these genes in more detail to see if they may have contributed to the intriguing avian phenotypes. We also note that there's a growing consensus that body size evolution, in particular miniaturization, was very important in the evolution of modern birds. You can see this paper from Mike Lee in Australia looking at the increasing miniaturization going on to the avian line, and we proposed that these two insulin factor genes were important in that transition. I want to switch now to talking about the evolution of flightlessness, and here we're looking at a group of very iconic species, the flightless peleognathe birds. This is I think, in order to really sort of call yourself an ornithologist these days, you've got to cut your teeth on this iconic group. They're extremely enigmatic. They've really frustrated systematists for over 150 years, and so we're going to sort of try our hand at them. They're, of course, well known for their dramatic morphologies associated with the loss of flight, including lots of limb development, loss of limb elements and digits, loss of the keel on the sternum, etc. And we've taken a genomic approach to begin to look at how these traits arise. The kicker here is that the phylogeny of this group has really changed in recent years, such that the flighted tinamous, which you can see right here, this is a paper from Mitchell et al., are now not hypothesized to be the sister group as they were thought to be for decades, but instead are nested within the ratites. And this, of course, leads us to the conclusion that flightlessness probably arose convergently along multiple lineages, whether it's these extinct lineages of moa and elephant bird, or the extant lineages of the other extant ratites. So this provides a very convenient framework for trying to pinpoint genes that may have contributed to the origin of flightlessness. So we've undertaken a small genome project. We've sequenced to high quality 10 paleognathe genomes. You probably can't read the statistics here, but suffice it to say that they're generally of very high quality. We've used a 2-library system using the all-paths assembly algorithm. What you can see here is the quality of our genomes as measured in the contig N50 length. These are here in red as compared to those published by the recent avian phylogenomics project. And you can see that our genomes compare very favorably to those of the Genome project, most of which have lower N50 contigs. And these are using approaches using far fewer libraries than they typically use. We've also been able to apply this approach to other genome projects and other systems we've been working on in the lab, such as the housefinch here and the storm petrel. Now, one of the things we've learned is that the sizes of these paleognathe genomes are actually smaller than are reported in the databases. You can see the reported values here in the red diamonds and our estimated values from the assembly sizes here. So this is intriguing. The ratite genomes do not seem to be as fat and lumbering as they've been suggested to be in the literature. That said, they are in fact larger than those of the tinamous, and so we're kind of curious what's driving that genome size increase. Now, surprisingly, we don't see much evidence for a proliferation of repetitive elements in these paleognate genomes. You can see here the percent that's masked by repeat masker. The flightless paleognates are here, and it's a very small percentage. You can see, in fact, most neognathous birds have a much higher percentage of repetitive elements, and so we're intrigued by this. And we're expecting to find a novel class of repetitive elements that isn't currently detected by current repeat masking protocols. We also are conducting a 46-way whole genome alignment among our 10 rat-type genomes, as well as other genomes from the Avian Phylogenomics project. This is an extremely computationally intensive project, but it's one that we're gradually working towards. And we're using a method called progressive cactus, which essentially moves down the tree, creates ancestral estimates of genomes, and then aligns those to each other in a progressive manner. What we can say is, having aligned the rat-type genomes to each other, we can begin to talk about the direction and rate of genome size evolution. And what's intriguing on this slide, you can see the key down here with increasing rates of genome size in the blues and decreasing rates in the browns. We're seeing some evidence for convergent increases in genome size across the different flightless lineages, such as ostrich, rhea, emus, etc. We're also seeing a dramatic decrease in size along the lineage leading to tinamous. This is probably part of the whole syndrome of features that really makes the tinamous genomes unique in terms of their base composition and presumably their structure as well. So we're moving towards a stage where we can essentially look at the origin of genes, candidate genes that are contributing to flightlessness, and also look at the regulatory elements surrounding those. And we're hoping to use this to identify regions that show unique patterns on flightless lineages, and ultimately to test these regions in a developmental context using experimental embryological approaches. So I'd like to thank my team here. This is a really fabulous team consisting of paleontologists and computational biologists, as well as bird watchers like myself, and I'd be happy to take any questions.